Hello everyone. Thank you for the opportunity of this presentation. My name is YiyIng Chen. I'm a postdoc fellow in Dr. Wenhua Li's lab in Academia Sinica in Taiwan. My topic today is homophilic ATP1A1 binding induced activin A secretion to promote EMT of tumor cells and myofibroblast activation. Tumor heterogeneity is the most distinct characteristic of cancer, which drive tumor progression, metastasis, and treatment resistance. The two major drivers of tumor heterogeneity include tumor intrinsic genetic instability and the influence from stroma cell in the tumor microenvironment. There's a common spatial heterogeneity in solid tumor that epithelial-mesenchymal transition, abbreviated as EMT, happens in the peripheral regions of tumor lobes, which lead to tumor invasion and metastasis. However, detailed mechanism of how stroma cells increase tumor spatial heterogeneity through triggering EMT remains unclear. Therefore, our study tried to understand how tumor stroma interaction regulates spatial heterogeneity of tumor and stroma cells in tumor boundary regions. Our studies focus on pancreatic ductal adenocarcinoma, abbreviated as PDAC, which is an important health issue. PDAC is characterized by desmoplasia, which contains a large amount of cancer-associated fibroblasts. As you can see, in human and mouse PDAC, specimen contained large amount of activated fibroblasts, also known as myofibroblasts, and stemmed by alpha-synuclein. Since fibroblast is the most common cell type in PDAC, I wonder how tumor fibroblast interaction regulates spatial heterogeneity in tumor boundary regions and whether tumor invasion can be enhanced. To explore the role of fibroblasts in human PDAC, I stent alpha-synuclein as shown by the green signal. Interestingly, alpha-synuclein positive fibroblasts were preferentially located in juxtatumoral regions as we analyzed the expression levels of alpha-synuclein compared 0 to 10 versus 10 to 20 microM distance from tumor margins. To further validate the clinical significance of juxtatumoral location of myofibroblasts, we collect a retrospective cohort containing stage 2 PDAC patients. The results show that the patients with high juxtatumoral alpha-synuclein expression was correlated to poor prognosis compared to patients with low juxtatumoral alpha-synuclein expression. Therefore, enrichment of alpha-synuclein positive fibroblasts in tumor stroma interface was correlated to poor clinical outcome. This result gives us a hint that contact-dependent interaction between tumor cells and fibroblasts may promote tumor malignancy. So first, I have to check whether heterocellular contact really happens between tumor cell and fibroblast. To answer this question, pancreatic tumor from transgenic mice were mechanically dissociated to cell clusters and stand for EpCAM and PDGF receptor alpha, which are membrane biomarker for epithelial cells and fibroblasts. Cell clusters with both EpCAM and PDGF receptor alpha expression were isolated by cell sorting and stand for CK19 as a biomarker for tumor or epithelial cell, and alpha-synuclein as a biomarker for fibroblast. Indeed, cell-cell contact between CK19 positive and alpha-synuclein positive cells were observed in vivo. Another evidence is that in a 3D-based co-culture system, using co-culturing of fibroblasts and tumor cells can result in heterocellular aggregation. Therefore, these results show that direct contact between tumor cell and fibroblasts can be observed. And the next question is whether direct tumor-fibroblast contact can promote tumor malignancy. To answer this question, we perform sphere assay and spheroid invasion assay. The results show that direct contact with fibroblasts compared to indirect co-culture or cancer-only condition can significantly increase the number of sphere and invasion. To explore the mechanism of direct tumor fibroblast contact-mediated tumor malignancy, RNA-seq analysis was performed to compare gene expression profile of tumor cell among monoculture, indirect co-culture, or direct co-culture conditions. The top canonical pathway regulated in tumor cells after direct contact with fibroblasts were related to fibroblast activation and regulation of EMT. The RNA-seq result was confirmed by the EMT signature genes, the mRNA level by qPCR, and the protein level by Western blot. Since EMT plasticity enhanced tumor progression and metastasis, we then compared the tumorigenic and colonization abilities between tumor cells and tumor fibroblast clusters. The results show that tumor fibroblast clusters significantly increase tumor growth and liver colonization. Therefore, direct contact with fibroblasts promote tumor malignancy by enhancing EMT. And the next question is, what's the mechanism of direct contact-mediated EMT of tumor cells? To discover the mechanism, I perform a cytokine array to measure cytokine secretion during co-culturing of BSPC3 cancer cells with PSC pancreatic stellate cell, which is fibroblast, in trans well with the indirect and direct contact conditions. And Activin A is the only cytokine have more than twofold increment upon direct co-culture. And indeed, Activin A can induce EMT and can induce sphere formation, and neutralize of Activin A can suppress EMT and suppress sphere formation. In addition, Activin A can activate fibroblasts measured by alpha-synuclein and collagen 1. Neutralize of Activin A can suppress fibroblast activation. Therefore, direct tumor fibroblast contact can trigger Activin A secretion, leading to EMT of tumor cells and myofibroblast activation. So the next question is, which cell type was the origin of Activin A during tumor fibroblast contact? To identify the origin of Activin A producing cells, we treat protein transport inhibitor to trap cytokine in the Golgi complex for immunofluorescence staining of Activin A. Accumulation of Activin A was significantly higher in direct co-culture fibroblasts than fibroblasts alone. To confirm the origin of Activin A producing cell in PDAC patients, mRNA of Activin A was detected as individual spots by in situ mRNA hybridization. Summary: enrichment of myofibroblasts in tumor stroma interface was correlated to poor clinical outcome. And upon direct contact with tumor cells, fibroblasts can secrete Activin A, and the juxtacrine Activin A will induce EMT change of tumor cell, and autocrine Activin A can induce fibroblast activation. To further dissect the mechanism for direct cell-cell contact to induce Activin A secretion, I had to identify the membrane protein for tumor fibroblast binding to induce Activin A production. To answer this question, I get a hint from a result that after I incubate fibroblasts with 2% paraformaldehyde fixed tumor cells, the Activin A can be induced. Consistently, Activin A can be induced in fibroblasts by tumor membrane proteins to differential extent. With this data, I have the opportunity to compare the differential membrane protein interactions formed between BSP3 SU.A6A6, which are Activin A inducing tumor cell lines, compared to MIAPA-CA2 and PANC1, which are cell line that is not able to induce Activin A in fibroblasts. And I labeled, to perform the experiment, I labeled the plasma membrane protein of tumor cell by a cross-linker containing biotin. And then I incubate this tumor membrane protein with fibroblasts. The fibroblasts are already treated with intracellular hydrogelation technique, which can make the cytosol and nucleus of the cell into a hydrogel-like condition. With the help of this technique, we can prevent the endocytosis of protein and then has the opportunity to enhance the purity of the plasma membrane protein for mass analysis. And then the tumor fibroblast membrane protein interaction were captured by monomeric Activin beads, and then the membrane protein were examined by SDS page. As you can see, a differential band around 130 kilodalton was specifically enriched in BSPC3 and SU.A6A6, which are tumor cells able to induce activin A in fibroblast. And so I cut this band for mass analysis. And among the 5 potential candidate proteins, knockdown of ATP1A1 can significantly suppress activin A secretion. Therefore, ATP1A1 of tumor cells is a mediator of contact-induced activin A secretion. So the next problem is, what's the binding partner of ATP1A1 in fibroblast? To perform the experiment, first I perform intracellular hydrogelation of pancreatic stellar cell, which is fibroblast. And then I label the plasma membrane protein with a cross-linker. And then I purify the plasma membrane protein and incubate the plasma membrane protein with agarose beads containing flake-only or flake-only control or ATP1A1 flake or another type of agarose beads as control. And the binding partner of ATP1A1 in fibroblast were identified. And I run the gel, and then I IB-Stravadin HRP, and can find a specific interacting protein noted in ATP1A1 flake group. And this protein cannot be stained by flake, so it's not a contamination. So I crop these bands for mass analysis and identify an ATP1A1 in fibroblast as its binding partner. And knockdown of ATP1A1 in fibroblasts also reduce activin A secretion. And as you can see in the immunofluorescence result, ATP1A1 is enriched in cell-cell contact region of fibroblasts between tumor cells and fibroblasts, and contribute by both tumor and fibroblast. And then I also confirm the result in human PDEX specimens using IHC staining of ATP1A1 and RNA-scope staining of activin A in a serial section. The results show that indeed ATP1A1 can be expressed in tumor stroma interface and correlated to surrounding activin A expression. So homophilic ATP1A1 interaction between tumor and fibroblast can activate activin A secretion from fibroblast. And then the next question is, how homophilic ATP1A1 interaction induce activin A specifically in fibroblast? To answer this question, I check the activin A expression. I check the ATP1A1 expression level first, and then I'll check the ATP1A1 induced activin A signaling in the next slide. So first I check the expression level. A tumor cell of fibroblast were monocultured or direct co-culture with paraformaldehyde fixed cells. And then the plasma membrane protein were isolated by intracellular hydrogelation. The results show that plasma membrane localized ATP1A1 in fibroblast was specifically increased upon direct contact with tumor cells. And this result also confirmed in the immunofluorescence staining result. Interestingly, the increment of membrane-localized ATP1A1 in fibroblasts depends on high ATP1A1 expression in tumor cells, since knockdown of ATP1A1 in tumor cells will abolish ATP1A1 expression in fibroblasts. Therefore, high ATP1A1 expression level in tumor cells can stabilize ATP1A1 in the plasma membrane of fibroblasts. And the next question is, what's the ATP1A1 associated signaling in fibroblasts for activin A production? To understand the gene expression of fibroblasts upon direct contact with ATP1A1 high-expressing tumor cells, we perform RNA-seq to compare fibroblasts in monoculture, indirect co-culture, or direct co-culture with ATP1A1 overexpressing BSPC3 and SU.A6A6 cells, or ATP1A1 low expression MIA-PACA2 and PANC1 cells. And the results show that exon guidance signaling and CRIB signaling in neurons are highly activated. These two signaling are both related to calcium. Consistently, intracellular calcium condition was specifically upregulated in fibroblasts during direct co-culture with ATP1A1 overexpressing tumor cells, while knockdown ATP1A1 in tumor cells diminished the intracellular calcium concentration. In addition, treatment with a calcium channel inhibitor, nifedipine, significantly suppressed activin A secretion. Therefore, ATP1A1 mediated calcium signaling can regulate activin A production. Previous results has shown that activin A production is regulated by NF-kappa B pathway in normal and cancer cells. Activation of NF-kappa B is measured by phospho-P65. Consistently, phospho-P65 was significantly increased in fibroblasts after direct contact with BSPC3 tumor cells through ATP1A1 interaction. In addition, NF-kappa B inhibitor can suppress activin A production. So these results suggest that direct contact with ATP1A1 overexpressing tumor cells trigger intracellular calcium oscillation that stimulate NF-kappa B signaling and activin A secretion in fibroblasts. Our study highlights the importance of tumor supporting niche modified by direct contact between ATP1A1 overexpressing tumor cells and fibroblasts. Homophilic ATP1A1 interaction facilitate heterotypic cell adhesion and directly regulate cytoplasmic signal transduction. This tumor promoting cell-cell interaction induce juxtacrine activin A to generate tumor cell with EMT plasticity and autocrine activin A for fibroblast self-activation. Tumor fibroblast cluster established at the tumor invasion front may survive to reach the general circulation and may facilitate collective invasion and metastasis of cancer cells. Blockage of direct contact between tumor cell and fibroblast may represent a promising therapeutic strategy for patients with PDAC. Thank you for your attention.